So next I want to talk about some important mechanisms cells use to ensure what we call the fidelity of DNA. So what is fidelity? Well, fidelity is trueness to something, okay? We think about it in relationships, we think about it in a variety of things, but fidelity as it relates to DNA relates to keeping things unchanged. So we want to avoid change in DNA in most cases because change oftentimes leads to problems. Mutations are changes that happen in the DNA, and mutations can of course give rise to things like death, like cancer, and all kinds of problems. And so cells have mechanisms in place to reduce the likelihood that problems will happen in the DNA. They're there to improve the fidelity of DNA replication. So I'm going to talk about really 4 different things. The first of these is one that is probably the simplest to understand, and that is a mechanism that's built into most, but not all, DNA polymerases. And this ability is something that we call proofreading. So proofreading is the ability of a DNA polymerase to not only synthesize a strand of DNA, as we see going on here, but also to frequently, not always, but frequently recognize when the polymerase has made an error, okay? So imagine that we have a polymerase that's going along and it has, for example, a G on the complementary strand, and instead of putting in a C, it puts in a T, or it could be an A that instead of putting in a T puts in a G or a C or something like that. All that matters for our purposes here is that there's a mismatch that's occurring. The mismatch meaning that we have the wrong base on the newly synthesized strand. Well, the DNA polymerases have additional enzymatic activities built into them. I mentioned, for example, that DNA polymerase 1 has a 5' to 3' exonuclease activity that helps the DNA polymerase 1 to remove RNA primers on the newly synthesized DNA. Well, it turns out that, of course, as I mentioned earlier, the DNA polymerase 1's 5' to 3' exonuclease activity is not found in other proteins, but the activity I'm getting ready to show you is found in most cellular DNA polymerases. And this activity is known as a 3' to 5', as the opposite orientation, a 3' to 5' exonuclease activity. So when I say exonuclease, what does that mean? Okay, a nuclease, first of all, is something that breaks down nucleic acid. So an exonuclease is something that breaks down a nucleic acid starting from the end. An exonuclease will start at the end. Okay, so the 5' to 3' exonuclease started at the 5' end and chewed inwards, breaking down that RNA and then replacing it by the polymerase with DNA. In the case of the proofreading activity, we have a 3' to 5' exonuclease. So that means it starts at the 3' end and moves in the 5' direction. Well, that turns out to be really useful for a DNA polymerase moving along. The DNA polymerase moves along, it's found and put in the wrong base. What happens when the wrong base gets put in? Well, it turns out that the wrong base doesn't quite fit in the duplex properly, so it makes a little bulge that looks like that. And that bulge is what's recognized by the polymerase. And the polymerase says, oops, made a mistake. Just like you might make a mistake if you were typing something and then you look down and you realize you've hit the wrong key. All right, what would you do? Well, you would backspace it out, right, to remove that key and then type in the right thing. Well, the 3' to 5' exonuclease of DNA polymerases is the backspace function. And it literally does that. It has been moving forward 5' to 3', it makes a mistake, oops, I made a mistake, I back up. And when it backs up, it removes that base with its exonuclease activity. That removal is occurring opposite to the way the polymerase was working. The polymerase was working 5' to 3', the exonuclease is moving backwards 3' to 5', and so that base gets removed as a consequence of that. And now that bad problem base has been removed, and the polymerase can now insert the proper base and move along. The remarkable thing is that this happens as rapidly as it does. So DNA polymerase 1 has proofreading activity, DNA polymerase 3 has proofreading activity, And remember that DNA polymerase III is moving at 1,000 nucleotides a second, okay? So it not only is moving rapidly this way, it's also proofreading what it's doing and moving back that way. Imagine that you were typing that fast and that you could recognize your errors and fix them and still keep your average rate of 1,000 nucleotides a second. So as I said, not all DNA polymerases contain proofreading activity. A notable DNA polymerase that does not contain this activity is the DNA polymerase that's found in a virus like HIV. HIV contains its own DNA polymerase that it codes for in its genome. And that DNA polymerase is known as reverse transcriptase. Reverse transcriptase, of course, is an enzyme that copies RNA and makes DNA. Reverse transcriptase does not contain any 3' to 5' exonuclease activity, which means that it doesn't proofread what it does. Well, if you don't proofread what you do, and you don't have a spell checker, for example, you look at what you've written, you're going to find there's going to be some mistakes in it. And that means that HIV's reverse transcriptase polymerase makes a lot more errors than a cellular polymerase. Well, that turns out to have a useful property for HIV because it means it's very commonly mutating, which means it's very rapidly evolving, which means that it can dodge various defenses that cells and doctors, for example, put up against it. So when we hear about HIV, a new strain of HIV evolving or a new strain appearing that's resistant to drugs that previously worked on HIV, that's happening because the polymerase is very rapidly mutating and getting around the older drugs that worked on it. Okay, so a second mechanism for maintaining the fidelity of DNA is known as mismatch repair. So there are 3 other repair systems that are found in E. coli cells and also in other cells that I want to talk about that are important for maintaining the fidelity of DNA, but I'm not going to go into them in this class in any significant depth. There's a common feature of all of them, and yes, there are many details about each of these that are different. I think to understand the bigger picture of these is the most important thing. So in mismatch repair, what do we have going on? Well, you might think from what I talked about with respect to proofreading that DNA polymerases never make errors. Well, it turns out that DNA polymerases aren't perfect. Proofreading improves the fidelity, that improves the replication of DNA accuracy considerably, but it doesn't make it perfect. Just like you find, for example, books that are published that have been proofread by many people, and there's still the occasional error that pops up. Well, that happens with DNA polymerases as well, and that type of problem is dealt with by this system that I'm going to describe to you called mismatch repair. So mismatch repair is a very important backup for proofreading that allows the cell to fix a mismatch that got missed by proofreading, okay? So from a publishing perspective, and we compare that to what I just mentioned before, we could say that, well, we've put out our first edition of our book, and oh my God, we've got this really embarrassing error on page 3. We're going to fix that error, and then we're going to put out a new edition of the book that won't have that error. So that's what's really happening with mismatch repair. Mismatch repair in this case has a base that didn't get, a base mismatch that didn't get caught by the DNA polymerase's proofreading activity, and now the duplex DNA has this error. As I noted during the replication, mismatches will have a little bit of a bulge. And so E. coli cells actually have a protein that recognizes that bulge. And it recognizes that bulge and says, okay, we've got a problem, we've got to fix it. Now, cells have proteins that recognize that bulge, bind to it, and then they do something interesting. You can see what's actually happening here, and I'm simplifying things quite a bit. But after recognizing the bulge, there are a couple of nicks that are put into one of the strands, and that an exonuclease activity removes one of the strands but leaves the other strand intact. If the strand that contained the wrong base is removed, then DNA polymerase can come along, fill in, And where there was an error before, now has been properly filled in. We now have a properly repaired DNA. The only key point of this slide, okay, that I haven't described that you need to take into consideration is the cell has to be able to know which strand had the error. Because if it doesn't know which strand had the error, then it's randomly going to fix the wrong strand half of the time and randomly fix the right strand the other half of the time. Well, that's not a very good way of fixing things. And it turns out that cells do, in fact, have a way of recognizing which strand is the newly synthesized strand and which strand is the one that was there before. Well, why would that tell? Well, remember that replication is making a new strand. And so if the proofreading is missing something, it's the new strand that will have the error. Okay? That's what the cell's concerned about. Does the new strand, was it properly copied or was it not properly copied? So the important point to recognize this, I'm not going to talk about the mechanism that the cell uses to do that, but the important point to recognize here is that the cell has to be able to recognize what was the strand that was being copied and what was the strand that's the new one. It's the new strand that gets excised and replaced. So that's mismatch repair. This strand, double strand that you see on the screen depicts a DNA that's been damaged in some way, okay? So this process I'm getting ready to describe to you is something called nucleotide excision repair, okay? Nucleotide excision repair is necessary for a couple of reasons. One is that DNA can be reacted with other compounds. So for example, in the case that we see here, here's a base that has had something humongous that's been attached to it. So for example, if you are a person who smokes or you breathe a lot of exhaust fumes from cars or other nasty things like that, these smokes that are there are very full of reactive molecules, very reactive molecules. And some of those molecules are very large in size, like this one. And some of those molecules can covalently bind to portions of DNA. So what's happened here is that this large molecule that might have been in cigarette smoke, for example, has covalently bound to a base in DNA. Well, as you can see, that's going to cause a problem. So first of all, we see a bulge, kind of like we talked about before with the mismatch. And that bulge is an indication that there's some kind of a problem, okay? The cell needs to be able to remove this, okay, as best it can. So I'm going to show you this. I'm going to show you another kind of bulge that can happen that's also treated by mismatch repair. So here's another bulge that can happen as a result of action. Get over here where I can see you. That can happen. So if we look on the left, for example, here's a normal unaltered strand of DNA. And over here, right beside my head, we can see that same strand of DNA where something has happened to it. So what are we looking at? So we're looking at one strand of DNA, and here are two thymines. So here are two thymines next to each other on the same strand. This is not on opposite strands, but this is on the same strand. In the presence of ultraviolet light, such as we would get if we went to a tanning booth, or if we spent too much time in the sun, what happens is that these thymines can have a covalent or two covalent bonds joining them. Well, that's a real problem. Notice that the normal one does not have bonds joining that, whereas after exposure to ultraviolet light in a tanning booth, those two thymines are joined together. The joining together of those two thymines creates what we call a thymine dimer. And thymine dimers are real problematic because they cause a bulge in the DNA, very much like we saw with the molecule that we added before. Well, either of these, whether we add something to a DNA or we create a thymine dimer, either of these cause problems in the DNA. The problems happen because when the DNA polymerase moves through there and tries to copy either the thymine dimer or the molecule that was sticking off of the base on the last slide, the polymerase is much more likely to make an error. Well, errors, of course, give rise to changes in the DNA, mutation, loss of fidelity. So cells have to have, or ideally have, a mechanism for dealing with these, okay? So what does that look like, all right? Well, there are a couple things that cells can do, for example, to fix that. This mechanism right here I'm showing on the screen is an interesting one that cells use to fix thymine dimers. So we can see here a DNA that has been exposed to ultraviolet light. We see the thymine dimer forming at this point. We see the bulge that's there. And E. coli cells have an interesting enzyme called photolyase. And so photolyase has a really cool property. It uses the energy of light. First of all, it recognizes and binds to the bulge. So we see the bulge right there. And when it binds to the bulge, it waits for another pulse of ultraviolet light. And what it does is it breaks the bond between the two thymines and restores the two thymines to their original state, thereby taking care of the problem. Okay? So photolyase is one way that cells have of fixing the problem. Photolyase is sort of an exception. Photolyase isn't nucleotide excision repair. I want to emphasize that. Although I'm talking about excision repair, this is not an excision repair process. This is an enzyme that's simply fixing a thymine dimer. There are other means of fixing thymine dimers that do require nucleotide excision repair. So let me move to those now. I just wanted to show you this one slide. So how does nucleotide excision repair work? Well, remember that we have either DNA adducts, those molecules, big molecules that are attached that cause the bulge, or we have thymine dimers that cause the bulge. So both of those are going to be fixed by this nucleotide excision repair mechanism. If you look at this, you'll see it looks kind of like what we saw with mismatch repair, although the enzymes themselves are different. First of all, we have a problem. In this case, we have a thymine dimer. We see the thymine dimer is recognized by proteins because of the fact that there's a bulge. And what does this stimulate? Well, it stimulates the removal of a strand. And in this case, it's quite clear where the problem is because we can see here, oh, here's the problem. We remove the top strand and we end up with this. So we've got a bottom strand that doesn't have that. And as a consequence, DNA polymerase comes along, it fills that in, and now we've got a DNA that's functional, has fidelity, whereas this one had problems. The issue was very easily fixed. You might say, well, why do cells have a mechanism for thymine dimers that involves nucleotide excision repair, and they have photolyase? Why doesn't it just have one? Okay. Well, first of all, you can see that nucleotide excision repair will work on something like thymine dimers, but it will also work on something that had a molecule attached there. Okay? So nucleotide excision repair can fix a variety of problems, not just one. Photolyase can only fix one problem. And the second is that the more repair mechanisms a cell has, the better able a cell is to maintain the fidelity of DNA. So these multiple mechanisms that I'm describing to you are a reminder of the importance of maintaining the fidelity of DNA. The last thing I want to say before I turn to the last mechanism, which is called base excision repair, is a reminder that even though our cells also have these mechanisms I'm talking about are in E. coli, our cells also have these repair mechanisms. It's important to recognize that just because we have them doesn't mean that, well, it's okay, I can go out, I can suntan as much as I want, I can go to a tanning booth and spend as much time as I want, or I can smoke cigarettes as much as I want and do all of these nasty things. The reality is that these are helps, but they're not solutions to preventing mutation. The more you go to a tanning booth, the more you smoke cigarettes, the more you breathe bad air or you drink bad water, the more likely you're going to overload all of these protective mechanisms and consequently favor mutation, and mutation leads to cancer. So that's really important to remember. So that's why I emphasize in class the importance of not smoking and not going to tanning booths, because they're pretty much equivalent in terms of nasty things. Well, the last mechanism I want to talk about is something called base excision repair. And you're going to see, again, it's kind of like the others, but it has an interesting wrinkle. Base excision repair is important because DNA itself is not completely chemically stable, all right? So, for example, what you see on the screen is a depiction of a reaction that can occur to the base cytosine. So cytosine has the structure, as you can see on the left, and as we look here, this is the base uracil. So what's happened in going from cytosine to uracil? Well, it turns out that cytosine is chemically unstable. Not real unstable, but at a low frequency, cytosine will deaminate, meaning it will lose this amine group here and have it replaced by an oxygen here. And when that happens, cytosine becomes uracil. Well, that is happening in our DNA all the time. We have cytosine being converted to uracil. And of course, you know DNA doesn't contain U, so the fact that DNA would have a U in it would indicate, oh, this process has happened, and that could be problematic if it's not fixed, because U forms base pairs just like T. Okay, well, what happens if a DNA that contains U gets replicated? Well, if it contains U, what will be put opposite this is an A, okay? Whereas the original C, if it got replicated, would have had a G. So we would have had a G being mutated to an A if this is not fixed. So the fixing of U's in DNA is very, very important because this process is occurring spontaneously all the time, okay? So how do cells do that? Well, they use this mechanism called base excision repair that is really interesting. Base excision repair, and this looks kind of like a complicated slide. The only point I want to make about base excision repair is that base excision repair involves an enzyme that recognizes that U, it binds to that DNA duplex, and then it does something really cool. It takes that U, and you can see the U right here in this little thing, and it flips it out of the duplex. So instead of the U being paired or mispaired with a nucleotide across it, it takes it and it twists it around and flips it out so that that U, instead of facing its complementary or the other base in the duplex, is now facing the enzyme itself. And so the enzyme clips off that U, okay? That's the base excision. It's removing that base. Well, what happens after it removes the base? Well, you're going to find this a little, maybe not surprising. Remember how we saw that one strand got removed and then the DNA polymerase came in and replaced that? That's exactly what happens here. So after the base gets removed, this strand that's on the left gets clipped, DNA polymerase comes in, replaces it, and everything is fine. So we saw that the 3 of the 4 mechanisms that I described, the mismatch repair, the nucleotide excision repair, and the base excision repair, all had a common feature. They all recognized the problem strand, they made cuts that removed part of that problem strand, and then DNA polymerase filled that in. They each had individual proteins that were different, and in the case of the this guy here, we can actually see a very different mechanism to start it, but in the end, they all worked in essentially the same way.